ma'am? Great. So it's an absolute pleasure to be here today, and I want to thank Craig and everybody at the museum for inviting me on this wonderful occasion. Now, I'm going to switch scales utterly and completely to the macro scale, or the biogeographic scale. So biogeographers and macroecologists spend a lot of time and considerable effort documenting patterns of diversity and trying to get a better handle on what types of things cause those patterns. And there's been a couple ways that we do this. One is using a phylogenetic perspective and looking at the history of a given group. And the other is where we get a lot of information on a ton of different species in the region and try to correlate richness, patterns, repetitive endemicity with either historical ideas of where vegetation was, current productivity, etc. And then the next sort of goal or ambition of people who do these sorts of research is to actually relate that to conservation. And can we actually use our techniques and our methods to help conservation biologists make decisions? So one of the exciting things that comes out of museums is this whole notion of all these point locality data, all this information of where things have actually been seen and where they're collected in these wonderful voucher specimens. And at the same time, there's been an absolute revolution in the availability of remote sensing data and environmental information. So the challenge then really is to put these different kinds of information together. And so I think a lot of crazy ideas come out when you're having a drink. Most people in science and in general, if you ever talk to someone who thinks they speak Spanish, if they talk about that and they say, yeah, you know, when I have a few drinks, man, I speak Spanish so much better. I've heard this before, oh man, when I have a few drinks, I can really dance. Biologists, I think, are the same way. They sit around and have a few drinks and they say, okay, well, all these data, I bet we can make really good maps with ad hoc, present-only data from museums. And so this was this kind of crazy idea, let's go out and prove to the world that we can do this. And this was sort of Craig Moran and I and Tom Peterson sitting around.
I wasn't having a drink.
And so the challenge really was this approach out there called distribution modeling. It's been around forever. It is basically what sort of we talk about when we talk about habitat-wildlife relationships. All of the USGS has been doing this stuff for years. But they have generally really good presence data, really good absence data, or somewhat good absence data, and they can put together information on where species are found and where they're not found, with a bunch of different kinds of environmental layers, often with a heavy emphasis on vegetation layers, and they can take these two kinds of information, where you have or don't have a species, and all of these different environmental variables, and come up with a pretty map, or a prediction of where that species might be. So just a couple of points on this notion is that before maybe 5-10 years ago, and even now, people are really skeptical of this approach, especially when you're taking museums with this ad hoc data that weren't collected in any systematic way. You guys go out to pretty places and you collect wonderful data, but you don't systematically always sample the environment. Even though some people, especially at the museums, did do beautiful systematic surveys, that's not the norm in museum data. So basically, our challenge was to actually say, can we do a good job of this with ad hoc presence-only data? And what came out of that work was a really fun paper with a ton of different collaborators that said, yes, some of these novel methods, and I won't go into details because nobody wants to hear methods talk, but some novel methods really do allow us to do a pretty good job of making distributions, of predicting species distributions with relatively ad hoc data. So this was an interesting result. This paper's been out about a year and a half. It's probably been cited a couple hundred times, or 100 times, a lot. So this is cool. People really want to make models. It's also really scary, because I'm not sure people they believe models without really thinking about them. But here are all these data that museums have provided. And I think this is another example where a place like Berkeley has been at the forefront of these kinds of analysis and pushing this integration with geographic information systems is just a really special place for that reason. And clearly there's a need and there's an interest in this kind of approach. This approach is species distribution modeling or ecological niche modeling, which I'm not sure Bernal would appreciate the way we use that, so it's called species distribution modeling. And this has given us a lot of opportunities. So what I want to do then is sort of talk about how we can use all of these existing data to look at Andean diversity. And I'll mention 3 different things in this talk. First, I'll explore some of the causes, some of the patterns of diversity in endemic birds in the Colombian Andes, some of the things we think might influence those patterns. I'll talk about the future persistence of endemic birds, and how we're looking at that. And then I'll talk a little bit, if there's time, about some of the threats to the persistence of economic frogs. So to introduce you to the Colombian Andes, this is an incredibly topographically complex area. There's a lot of bird species here, and there's a lot of endemic species, about 220, where endemicity is defined by having a range of about 50,000 square kilometers. There's a couple endemic birds. What you see in the Andes, in the Colombian Andes, is a lot of topographic complexity. You have these 3 cordilleras, which vary in age, this being the youngest, with its last big mountain rising period is about 2.7 to 0.6 million years ago. And these central cordillera is the oldest, and this being of unknown age, but also likely quite old. And you have these valleys in here, and lots of isolation. And you also, when you actually ask how, the other thing is how similar are environments across these areas. This is just environment characterizing all the environments that exist within the Western Andes and the Pacific slope, and asking where else those environmental conditions occur. Same things for the Central Andes and the Eastern Andes. And a really quick thing to see there is a lot of blue in all the cordilleras, regardless of where the environmental space is sort of extracted from, which means the environment across the Andes is quite similar. So there's a lot of repetition of environments, potentially at this broad scale, and also a lot of topographic complexity. So how does this all go together? How can we put this together and try to understand diversity? And I just wanted to put that up there for people who don't realize how complicated the Andes are. This is just nature serve maps or range maps from the species group Galeria in Colombia. You see all kinds of different distributions. So you have species on one mountain range and not others, etc. And this is some work that's been done by Gustavo Catán, where they're just actually comparing species similarity across these different bioregions, these different slopes. And they basically find that you have a lot of similarity on the inner Andean slopes here, in the greens and the blues, the purple slope here, the Pacific slope being different. Again, this western slope being slightly different. Lots of sort of different species up here, lots of endemism, both at the whole region as well as within these specific slopes. So what we want to do now is throw as much information as we possibly can at this question to better define these patterns. So far, all we have are some range maps, which are sort of at the level of the nature serve range map, and just expert opinion maps. And we have these sort of broad biogeographic types of information about how similar different types of the range are. So can we do better, and how can museum data specifically help us do better? So this is actually a project that was Project Biomap, and it used museum data, information from some of the local conservation organizations, and personal communications literature, getting together as much as we possibly could on all endemic, that's 218 species, and threatened birds in Colombia. And you can see these are the points. We've also tried to gather points from close by regions of Ecuador and Peru and Venezuela to get a more complete understanding of the environmental conditions species have. That, after a huge effort, this resulted in about 4,000 unique localities across all these species, with most species having, here's the mean or the median, about 11, and a lot of species without quite enough information. But quite a few species with enough information to try to do this distribution modeling and address the more biogeographic questions with these data. And another really exciting thing we did, which is really simple, is we just threw all of our maps up onto the web in an interface where people could go in and look at them and say, this is a good map, this point is insane, we missed this point here, I know it's been there, I've been surveying that area. And we got all of this information and we sort of weeded it out, but 14 really good ornithologists from Colombia put in their information, tons of comments, new localities, and we just threw that up on the website for people. And this is kind of exciting in two ways. One is it certainly helps us clean up our data. It helps us actually get some more data. And it helps, I hope, eventually, when we actually do stuff with these maps and try to get people excited about using them, to feel like they've had some buy-in to the process. So once we do all that and we map all of the different species, all of these endemic species I'm going to concentrate on now, you see you have a lot of endemic species in this Pacific Cordillera. We know this. This was also very similar to what Gustavo Catani found out. But you see we have a much finer spatial pattern of where the species actually are high. We did all of our modeling using maximum entropy models and current data. And we had to actually cut the models to deal with this problem of overprediction. And I'm happy to talk with people about what sorts of methodologies we explored to do that with. What you see is the highest richness here on the eastern slope. And so, well, why is that? What are some of the hypotheses? And can we use modeling to start to address some of the hypotheses of why you have such high richness in endemics here? Well, a couple of hypotheses you can come up with. Well, it's older. Bigger. Well, it's older. It's not older than the Central Cordillera. It's not necessarily bigger than these other Cordilleras. Not at all. It's got a source of potentially birds coming in across this isthmus. But when you actually look at the taxonomic affiliation of the birds, there's not hugely more birds from this sort of or predicted to originate in Central America here. So maybe there's something to that, but it's not extremely compelling. So, but one of the things that we could show that is really kind of cool and compelling is if you now take all of these maps and all this information and say, based on current environmental relationships between where species are and the climate, where would they likely have the last glacial maximum? And this is just one period in the past, and this is quite preliminary. And you see the coast is different here, which is why there's a whole lot of purple swimming in what looks like the ocean. But basically, we took the predicted endemism in the present, subtracted the predicted endemism in the past, and anything that's green here has greater endemism today, and anything that's purple here has greater endemism in the past than it does today, right? And so all of these purple areas then represent refugia. And this is particularly notable here in the Chocó region of Colombia, and that extends down into Ecuador. And this has actually been a region that has been hypothesized to be an important refuge for birds in this part of the Andes, part of the world. And the models really sort of show that that may well have been the case, and that may well be contributing to the fact that this cordillera has such high species richness. So the models are putting together this information, are starting to help us address some hypotheses about patterns of endemism. The other thing that we've been doing is actually saying, okay, we've got these broad patterns. Now, if we look within cordilleras, and specifically at range limits within cordilleras, can we then get even more insight about what might be going on? So range limits, determining what influences range limits is incredibly interesting, and it can help you predict or understand what kind of biotic or abiotic factors might occur for your range limits to define where they are, what kind of dispersal limitation might have occurred both currently or in the past. And then there might be areas where tons of ranges meet, and that's sometimes been called a suture zone. And if that's the case, you have lots of range limits in a given spot, you might sort of suggest that there's some similar underlying process that's actually causing that. And so one of the fun things that we can do environmentally. This is sort of the state-of-the-art distribution of the species. And you can see you have a lot of prediction beyond either where the point localities are or where the little ridgely range map is. So if we take this range now, this is just zooming into the southern limit, we can say, at least within this bioregion, the Western Cordillera, it peters off right here, right? And here, in the north, there's a break here, there's a break here, there's a break here, right? So we can then just sort of map where we think these range limits are. So here, to the best of our knowledge of where the points are and where people have seen it, we put a line there. That's the range limit in the south. And here's the northern range limit. Could be here, maybe there. It's a little tricky because we only have points down here. So we can do sensitivity analysis and try to look at both of those possibilities. Okay, so we've done this now for the 105 species for which we actually have a relatively decent amount of data. And what you see here are the point localities that we have, and then all of these range limits. And they're just colored different colors because I did it separately for different bioregions, for different cordilleras. And basically, for each range limit we add, we just say, is there any overprediction beyond that range limit? Are these must have been anywhere? And if there's overprediction, when there's overprediction beyond the range limit, but within the same biogeographic region. There's often overprediction to another biogeographic region, but we know there's really substantial barriers there. So if we want to understand potentially what is important driving diversity within a biogeographic region, we need to sort of study them one by one. So why are there more species potentially in the Western Cordillera? Okay, so this is just an example of what that looks like. Here's a region where a ton of species, or 12 species, had a break. And this is now just for the single bioregion. And you see very little overprediction beyond this particular spot. For these species, each line represents a species. But the other thing you see is a lot of overprediction into the other cordilleras, which we expect when we think about that overall similarity of environmental space across the entire region. So this is basically what the methods look like, what we did. And so now what you see here is just a composite of the range limits for the Pacific Cordillera. And you see on this side all those range limits which have prediction beyond the limit. So there's a range limit here, and it predicts beyond it, either the south or the north. So the range limit doesn't stop where the range limit is within where the species could actually exist. So the species can live in regions, or in conditions are good for the species beyond its range limit. That's basically what that's saying. This is when there's no prediction beyond the limit. And so basically, this is the limit for a group of species, and there's no prediction beyond that. So the limit seems to correspond to the petering off suitable environmental conditions. And so a couple things you can see here is, in this region you have what looks like some regions where a ton of species stop up here in the north. And these actually correspond to some really big valleys up in the north here. So it looks like what's happening, there's a lot of species that respond, some concordance in the patterns there, and a lot of overprediction, suggesting that it's really isolation that's causing that limit, dispersal limitation by those species, essentially. In contrast, if we look at this place where there's little or no prediction beyond, you don't see much going on in the north. We just saw there's a lot of overprediction beyond the range there. But you see a lot happening in the south, where basically the suitable habitat is just petering off as you go south to these different species. And the range limit seems to be defined by the availability of suitable habitat. And so now this is just to contrast this Cordillera with the Eastern Cordillera. And what you see here is there's very little overprediction in the Eastern Cordillera at all. So this would be prediction beyond the limit, just in a few cases. And there's little or no, this is the case where there's little or no prediction beyond the limit. And you don't also see much clustering here. This sort of seems to occur in this broad region and this broad region, but no specific place where all these species seem to be stopping. Not nearly as clear as in the other cordillera. So these are some somewhat preliminary analysis, but what they can start to tell us is that species range limits cluster in certain areas in some parts of the Pacific, especially, and a little bit in the eastern Cordilleras, and at least some of those northern ones seem to correspond to barriers, to really big valleys. Many range limits also seem to be sort of randomly located, suggesting species-specific responses to the environment actually causing those range limits, or defining those range limits, and range limits in the western and Pacific Cordillera, the Pacific Cordillera, seem to be more influenced by big barriers, which might help to explain why there's more species in the Western Cordillera. Okay, so that's just an example of how we can use distribution modeling to try to better understand range limits and better understand what might be causing high patterns of diversity, patterns that we see of diversity in the Andes. I'm just going to go through this next part a little quickly. What we've been doing is actually saying, well, we have all this information. Can we use it to actually try to understand how climate change might influence species richness patterns? So here's our current species richness map, and this is basically, if you project into the future, what species richness looks like. It goes down quite dramatically, and the question is, where does it go down? And what you see here are change hotspots between endemics and threatened species. So we looked at both the endemics, about 200 species, and threatened, about 87. And what you see is along the eastern Cordillera, you have the western Cordillera, you have a really strong sort of hotspot where you would expect a lot of lower predictions across a lot of different species. And this is just what's going to happen to range sizes based on these predictions. And there's different types of predictions that we've done, and you can see that range size is predicted to decrease, as one might expect in the mountainous area. And some ranges do decrease, that's just the amount that they increase. And I won't go through all this. These are some of the species that are condemned by the models to extinction. All these species live on isolated mountain massifs and mountaintops. And then we can take all this information and go into a conservation algorithm and ask, what are the areas that if we lost, what are the areas that are just very important for biodiversity? And we do something called irreplaceability to measure that. And so basically, here's what areas are really important for conservation currently. Here are what areas that would be really important for conservation in the future. And then we ask, what is the change in the areas that are really important for conservation between the current sort of patterns based on current patterns of species richness and based on future patterns of species richness, and we can come up with sort of hot spots of irreplaceability. Those regions that aren't particularly important now, but will be particularly important in the future. And we can identify those. And basically, new irreplaceable areas, which are in green here, are moving up the mountains, something that we would predict. And then you just ask how much of the current biodiversity is protected in current protected areas, and how much is going to be protected in protected areas in the future in Colombia. And you see that basically 60% are currently protected within the current protected areas. And in the future, we expect that number to go down to about 42%. Okay, and I won't talk about this because I'm out of time. Well, I wonder. So another thing that we've been having a lot of fun with is trying to identify threats for frogs and looking at a bunch of different things at the same time. Land use change, habitat change, blah, blah, blah. All the different hypotheses you can come up with that you could address with the GIS. And I'm just going to mention these here. This is something people know about. This is from pounds. For pretty much the optimal range for temperature should be between this temperature range. When we mapped that, and this is a frog that people might know, an Adelophus species that's very common. This is a distribution based on museum records, the last one of which is from 1982. This is its species distribution model. This is the range of temperature between 17 and 25 degrees Celsius. There is no correspondence in Ecuador. So what we did is come up with an ecological niche hypothesis for putrid in Ecuador. And here it is. It's not based on more than about 8 or 9 actual samples of putrid, so it's a little preliminary. But what you see, which is really interesting, is we sort of define this environmental space of Chytrid and the environmental space of the declining frogs. And I think most excitingly is if we actually take out elevation of temperature from this analysis in a sense. Because we know temperature is important. It's very clear, if I had time to show you those axes, that temperature is important for where Chytrid is most strong, or most related to where the critically endangered frogs are. If you take temperature out completely, which is what we see here, we actually even get a tighter relationship with, this is the Chytrid space, this is the critically endangered frogs, and they tend to be, what's now defining this overlap is actually precipitation, and precipitation during the coldest period. So areas where you have less precipitation, when it's cold, seem to have more species, sort of more of a stronger overlap with Chytrid. So this is what this analysis is telling us. And so by doing this, sort of, this is what's defining those PCA axes, basically. So by doing this sort of an overlay between where Chytrid is now and where endangered species of frogs are now, you can start to ask, at this broad scale, what might be some of the climatic variables that are particularly important or might be related, and then we can actually go and do some of the experiments that Rachel's been talking about, what do I say, Erica talked about, and actually test some of these hypotheses based on real data. Okay, or based on some hypotheses. So I just want to thank all the tons of museums that contributed data to this research, and without the museums and their wonderful databases, none of this would be done. And with that,